Okay, let's label this animal cell together so that you can color it correctly when you color it today. Alright, so first off, let's label the cell membrane. Remember, the cell membrane is what surrounds the outside of the cell. So this outer part here should be the cell membrane. You need to be labeling this on your paper as you are watching the video. Okay, next we need to label the cytoplasm. Remember, the cytoplasm is the gel-like fluid inside the cell that surrounds all the organelles. So this would be our cytoplasm. Next, we're going to label the nucleoplasm, or this is the fluid inside of our nucleus. This is this white area inside of our nucleus here called the nucleoplasm. The nuclear membrane, or the nuclear envelope, is what surrounds our nucleus. So this is going to be the little rim around the nucleus that you can see here. So that little rim is called the nuclear membrane. Next, we need to label the nucleolus. This is where ribosomes are produced, and this is found inside of the nucleus. So this is going to be this circle inside of our nucleus here. This is the nucleolus. Alright, Golgi apparatus. Remember, this is our packaging, sorting, and shipping center of the cell. One way you can tell the Golgi apparatus apart from the ER is that it's going to be further away from the nucleus, whereas the ER is always going to be right around the nucleus. So that means this flattened stack looking of pancakes over here is going to be our Golgi apparatus. Okay, the flagella is a long whip-like tail. It's going to help the cell to move, so that's the tail up here. The rough ER, remember, this is going to be bumpy. It has the ribosomes attached to the surface, and this is what makes it look bumpy. And so the rough endoplasmic reticulum is going to be this area, this here, that has the ribosomes attached. So that's our rough ER. Okay, the smooth ER is going to be over here. This looks like the rough ER, but it doesn't have ribosomes. So here's our smooth ER. Remember, your ER is always right around the nucleus. Next, we have mitochondria. These kind of look like little sausages inside of the cell. And so we have 3 of them in this cell. This right here would be our mitochondria. The lysosomes, these are these small circles that are going to break down the waste inside the cell. So this circle here would be one of the lysosomes. Our microtubules, these make up part of the cytoskeleton, and they look like these chopsticks inside of the cell. This is a 2D model, so they don't stretch all the way through the cell like they normally would, because otherwise we wouldn't be able to see any of the other organelles. And lastly, we need to label our ribosomes. These can be found in 2 different places, either attached to our rough ER or free-floating in the cytoplasm. So this is one ribosome. These are ribosomes here. And these on the rough ER are also ribosomes. So when you're coloring the rough ER, make sure you do not color the little circles dark blue. Make sure you color them red like they're supposed to. Okay, so let's scroll down and look at the plant cell and talk about the differences between the plant cell and the animal cell. Let's see if I can get this to scroll down here. Okay, so looking here at our plant cell, there are some differences in the plant cell versus the animal cell. And so plant cells, remember, always have a cell wall. So in the plant cell, this outer layer here would be the cell wall, which provides the rigid structure for the cell. the inner membrane here, this little rim, this is a cell membrane, so this is what you should be coloring orange. On the plant cell, they also have a large central vacuole. Okay, animal cells also have vacuole. It wasn't pictured in our coloring on page on the front of the page, but plant cells have a large vacuole, remember, to store extra water for photosynthesis. Speaking of photosynthesis, plant cells are also going to have chloroplasts, which are going to be these structures here, which are going to be responsible for doing photosynthesis for the plant cell. Okay, the rest of them, the structures are repeated from the front, so you should be able to figure out the rest of these structures and color this. It must be colored and completed and turned in by the end of class.